Today we're going to explore one of the most elegant techniques in modern chemical engineering, using microfluidic devices to create monodisperse emulsions. This is a technology that's revolutionizing drug delivery, food science, and materials synthesis. Let's start with the basics. What exactly is an emulsion? An emulsion is a mixture of two immiscible liquids, meaning liquids that don't naturally mix together, like oil and water. One liquid forms tiny droplets that are dispersed throughout the other liquid, which we call the continuous phase. Here's a visual representation. The orange spheres represent oil droplets dispersed in a water continuous phase. Common examples include milk, mayonnaise, and salad dressings. Now here's the challenge. How do we typically make emulsions? Traditional methods involve vigorous mixing or using high shear homogenizers. You're essentially using brute force to break up the dispersed phase into droplets. The problem is that these methods produce a wide distribution of droplet sizes. Some droplets are huge, some are tiny, and everything in between. This is what we call polydisperse. Notice how the droplets vary dramatically in size. This inconsistency is a major problem in many applications. You might ask, why does it matter if droplets have different sizes? Let me give you 3 compelling reasons. First, let's consider drug delivery. Imagine you're encapsulating a chemotherapy drug in lipid droplets for targeted cancer treatment. If your droplets have different sizes, each droplet contains a different amount of drug. This means unpredictable dosing. More importantly, different sized particles circulate differently in the bloodstream. Small particles may be cleared by the kidneys, while large ones get trapped in the liver or spleen. The circulation time is inversely proportional to diameter. Larger particles are cleared faster by the reticuloendothelial system. Research shows that the optimal size for tumor targeting through the enhanced permeability and retention effect is between 100 and 200 nanometers. If your size distribution is too wide, most droplets won't be in this therapeutic window. Now let's look at food science. Consider making a premium salad dressing or a gourmet sauce. Uniform droplet size give you a consistent, creamy mouthfeel. Polydisperse emulsions feel grainy or inconsistent on the tongue. Monodisperse emulsions are also more stable against creaming and coalescence. Creaming is when droplets rise to the top, and coalescence is when droplets merge together. The creaming velocity follows Stokes' law. It's proportional to the radius squared, so larger droplets cream much faster than smaller ones. If you have a wide size distribution, you get rapid phase separation. Finally, let's discuss material synthesis. Emulsions are used as templates to create porous materials, microparticles, and nanostructured composites. The droplet size directly determines the pore size or particle size in your final material. If you want controlled porosity for catalysis or controlled release, you need monodisperse droplets. For example, polymer microspheres used in chromatography columns must have extremely narrow size distributions. Otherwise, you get poor separation efficiency and broad peaks. So how do we solve this problem? The answer is microfluidics. Microfluidics is the science and technology of manipulating fluids at the microscale, typically in channels with dimensions from 1 to hundreds of micrometers. The key advantage for emulsion generation is precise, deterministic control over individual droplet formation. Each droplet is formed identically. There are 3 main microfluidic geometries used for droplet generation. Let me show you each one. The first and simplest is the T-junction geometry. In a T-junction, the dispersed phase flows perpendicular to the continuous phase. At the junction, the shear forces from the continuous phase pinch off uniform droplets. The droplets are all the same size because the forces are identical for each formation event. The second geometry is flow focusing, which is probably the most widely used. In flow focusing, the continuous phase flows from 2 sides and squeezes the dispersed phase through a narrow orifice. This creates even more uniform droplets and allows for smaller sizes, down to a few micrometers or even nanometers. The third geometry is co-flowing, which is excellent for creating core shell structures. In co-flowing, both phases flow in the same direction through coaxial capillaries. The inner fluid breaks up into droplets due to Rayleigh plateau instability. This is particularly useful for making double emulsions or encapsulated structures. Now let's understand the physics of droplet formation. What forces are at play here? Droplet formation is governed by a competition between interfacial tension, which wants to minimize surface area, and viscous drag forces from the continuous phase, which want to pull the droplet away. We quantify this with the capillary number. Ca equals mu versus over gamma, where mu is viscosity, v is velocity, and gamma is interfacial tension. At low capillary numbers, interfacial tension dominates, and you get larger, more spherical droplets. At high capillary numbers, viscous forces dominate, and you can generate much smaller droplets. The droplet diameter is proportional to the flow rate ratio of dispersed to continuous phase. This is your primary control parameter. Let me show you a practical back-of-the-envelope calculation. Suppose we want to make 50 micrometer diameter droplets for a food emulsion. How do we design our system? Let's say we set the continuous phase flow rate at 100 microliters per minute. This is a typical value for lab-scale microfluidics. We want droplets with diameter 50 micrometers. The volume of each droplet is 4/3 pi r cubed. With radius 25 micrometers, that gives us about 65 picoliters per droplet. That's 6.5 times 10 to the negative 11 liters. Now let's say we want to generate 100 droplets per second. That's a reasonable generation rate. The dispersed phase flow rate needs to be 100 times the single droplet volume per second. That's 6.5 times 10 to the negative 9 liters per second. Converting to microliters per minute, we get about 0.39 microliters per minute. So our flow rate ratio is about 1 to 250. This ratio gives us precise control. If we want smaller droplets, we decrease the dispersed phase flow rate or increase the continuous phase flow rate. How do we quantify monodispersity? We use the coefficient of variation. The coefficient of variation equals the standard deviation divided by the mean diameter times 100%. For traditional bulk emulsification methods, the CV is typically 20 to 50%. That's extremely polydisperse. Microfluidic methods can achieve CV values less than 5% and often less than 2%. That's remarkably monodisperse. Here's a comparison. The red curve shows a broad size distribution from traditional methods. The green curve shows the sharp, narrow distribution from microfluidics. The difference is dramatic. Let me give you a detailed application example. Suppose we're developing a liposomal formulation of doxorubicin for cancer treatment. Doxorubicin is a powerful chemotherapy agent, but it's very toxic to healthy tissues, especially the heart. We want to encapsulate it in liposomes for targeted delivery to tumors. Based on the enhanced permeability and retention effect, we want our liposomes to be about 120 nanometers in diameter. This size can extravasate through leaky tumor vasculature. If we use a polydisperse formulation, particles smaller than 50 nanometers get cleared by the kidneys before reaching the tumor. Particles larger than 300 nanometers get trapped in the liver and spleen. The therapeutic efficacy is proportional to the fraction of particles in the optimal size range. With a polydisperse formulation, maybe only 30% are in the right range. With microfluidics, we can get over 90% of particles in the optimal size range. This dramatically improves therapeutic efficacy while reducing side effects. Clinical trials of monodisperse liposomal formulations have shown up to twofold improvements in tumor accumulation compared to polydisperse formulations. Now let's look at a food science application: flavor encapsulation. Many flavor compounds are volatile and degrade quickly when exposed to air, light, or heat. We want to encapsulate them in oil droplets suspended in a gelatin matrix. For optimal flavor release and mouthfeel, we want droplets between 10 and 20 micrometers. Too small, and the flavor releases too quickly. Too large, and it tastes oily. With microfluidic generation, we can precisely control the droplet size, and therefore, the flavor release kinetics. All droplets release their contents at the same rate, giving consistent sensory experience. The release time is proportional to diameter squared divided by the diffusion coefficient. So even small variations in diameter lead to large variations in release time. Monodispersity is crucial. Finally, let's explore a material synthesis application, creating porous polymers for catalysis. In emulsion templating, we create an emulsion where the droplets act as templates. We polymerize the continuous phase, then remove the droplets, leaving behind pores. The pore size in the final material directly equals the original droplet size. If we want controlled pore size for selective catalysis, we absolutely need monodisperse droplets. For example, if we're immobilizing enzymes, we might want 5 micrometer pores. These are large enough for substrate molecules to diffuse in, but small enough to retain the enzymes. The porosity of the final material is controlled by the volume fraction of the emulsion phase. We can calculate the porosity using this equation, where phi emulsion is the volume fraction of droplets. Monodisperse pores ensure consistent mass transfer throughout the material. Every region has the same transport properties, leading to uniform catalytic activity. Now let me discuss some practical considerations and challenges when working with microfluidic emulsion generation. The first challenge is throughput. Microfluidic devices typically produce droplets at rates of tens to thousands per second. For industrial applications, this might not be enough. The solution is parallelization. We can fabricate arrays with hundreds or thousands of identical nozzles operating simultaneously. This scales up production while maintaining monodispersity. Modern parallelized systems can produce over 1 liter per hour of monodisperse emulsion, which is approaching industrial relevance. The second challenge is device fabrication. Most microfluidic devices are made using soft lithography with PDMS, which is time-consuming and requires clean room facilities. Emerging solutions include 3D printing of microfluidic devices and injection molding for mass production. These approaches dramatically reduce cost and fabrication time. The third challenge is selecting the right surfactant. You need a surfactant that stabilizes the droplets, but is also compatible with your application. For drug delivery, you need biocompatible surfactants like phospholipids or pegylated lipids. For food applications, you need food-grade emulsifiers like lecithin or proteins. Let me introduce an advanced technique: creating double emulsions, which are emulsions within emulsions. A double emulsion is water in water, or WOW. You have water droplets inside an oil shell, which is itself suspended in water. These are incredibly useful for controlled release applications. Double emulsions can protect sensitive compounds from the external environment. The inner aqueous phase can contain proteins or peptides, protected by the oil shell. Microfluidics allows you to create these complex structures with precise control over both the inner droplet size and the shell thickness. This is nearly impossible with traditional methods. Let me quantify the benefits of microfluidic emulsions compared to traditional methods across our 3 application areas. In drug delivery, microfluidic monodisperse formulations show 2 to 3-fold higher tumor accumulation compared to polydisperse conventional formulations. In food science, monodisperse emulsions show 5 to 10 times longer shelf stability against coalescence and creaming compared to conventional emulsions. In materials synthesis, monodisperse pore structures can show up to 100-fold improvement in selectivity for size-dependent separations or reactions. What does the future hold? The cutting edge is moving towards smart, responsive emulsions. Researchers are developing stimulus-responsive materials where droplets release their contents in response to specific triggers like pH, temperature, or light. For example, pH-responsive drug carriers that release their payload only in the acidic tumor microenvironment. The monodispersity ensures all droplets respond at the same pH threshold. Another exciting direction is integrating microfluidics with artificial intelligence for real-time optimization of droplet generation parameters. Let me leave you with a final calculation about economic impact. Consider a pharmaceutical company developing a new liposomal drug formulation. With conventional bulk methods, maybe only 30% of particles are in the optimal size range. The other 70% are wasted material. With microfluidics, 90% are in the optimal range. That's a 3-fold improvement in material efficiency. For an expensive drug like doxorubicin that costs thousands of dollars per gram, this is enormous cost savings. Additionally, the improved efficacy means you can use half the dose to achieve the same therapeutic effect. Combined with the material efficiency, this could reduce manufacturing costs by 6-fold. Let me summarize why monodispersity matters so much. In medicine, monodispersity gives you predictable pharmacokinetics and biodistribution. Every particle behaves identically in the body. In food, it ensures consistent quality, sensory properties, and long-term stability. In materials, it enables precise control over properties like porosity, permeability, and catalytic activity. And microfluidics is the key enabling technology that makes true monodispersity achievable at scale. The field of microfluidic emulsion generation is rapidly maturing from laboratory curiosity to industrial reality. Commercial systems are now available that make this technology accessible for routine use in research and development. The challenge now is scaling up to true manufacturing volumes while maintaining the exquisite control that microfluidics provides. We're seeing integration with continuous manufacturing paradigms, particularly in pharmaceutical production, where the FDA is encouraging continuous processing. Microfluidic generation of monodisperse emulsions represents a transformative technology that bridges fundamental physics, engineering design, and practical applications across diverse fields. We've seen how understanding the fundamental physics of droplet formation enables precise engineering that leads to real clinical and commercial impact. The power lies in controlling something as seemingly simple as size distributions, which cascades into dramatic improvements in performance. This is engineering at its finest, taking fundamental scientific principles and translating them into practical solutions that improve human health, food quality, and materials performance.